Yeah, let's do it.
Perfect. Hello, everyone. Welcome to today's town hall on July 20th, 2025. We are excited to be presenting on clinical trial research basics. And today's topic will be a little bit different. We're going to be going through something called a fireside chat, where we'll be doing a little bit of role playing. Karen Ho will be representing a industry or drug developing sponsor, and she'll be able to provide the perspectives of a drug development company on how they think about clinical trials and research. And there are some important elements that we'll highlight, about that role and that responsibility, and that's that drug developers cannot directly talk to patients. They want to care for the patient and create an opportunity to become involved with research, but they have to go through third parties in order to interact with patients. I'll be playing the role of a clinician. I'm doing the hand waving now that I am not a clinician, so I'll be doing the full hand waving there. But talking about how your clinician should be interacting with you in regards to clinical trial research, the advice that they should be giving, and different, points of consideration for us as caregivers when we're talking to our clinician. And Morgan will be playing the role of a caregiver on our fireside chat here. Our agenda for today is we'll go through what is clinical research and why does it matter, clinical trial stages and types, trial study designs, drug modalities, trial eligibility, trial participation, data collection, hypothetical clinical trial experience, and then community resources we have at Families SCN2A to provide to you all as caregivers as we, think about whether clinical trials are right for our family, and if so, how do we best do our diligence to make sure this is the right opportunity for our family. So again, fireside chat. We have three different roles here. Karen playing the drug developer, I'll be playing the clinician, and Morgan will be playing the patient caregiver. So what is clinical research and why does it matter? Karen, I will have you lead this as a drug development sponsor.
Sure. So, as it says, clinical research is a kind of scientific approach to healthcare, and we are trying to study how health and disease presents itself in humans, and we also want to aim to develop new and improved ways to treat those diseases. Um, Clinical research, as you know, is clinical to advance medical knowledge and develop new treatments and improve patient care. And in fact, we chose this topic for tonight because of a poll that Leah had posted on Facebook. And the vast majority of respondents of that poll, you all, told us that you want to learn more about clinical trials, clinical trial opportunities, and clinical research in general. So that's why we're here today talking about this topic.
Perfect. Thank you, Karen.
So as a caregiver, I have a question, as you just wonderfully introduced that. Does clinical research really involve the testing of new medications, or does it include other things like scans, like MRI or whole exome sequencing? I'm just hoping if you could clarify that, and maybe the next slide will help.
Absolutely. So you can think of clinical trials as oh, did you want me to take this, Sean, or did you want to take it? Because this is your slide, but I'll answer the general question. You can think of clinical research as interventional or non-interventional. So some of the things that you mentioned, Morgan, like scans, things that we use, tests, blood tests that might be used to track the trajectory of a disease and describe that disease using these particular experiments and assays are not necessarily interventional. So they don't necessarily involve a drug that is used to modify these measurements that were taken. In fact, we just want to learn about the progression of that disease as that person goes through it. And those kinds of studies are considered non-interventional because there's no intervention that's being tested. However, if a medical device or drug are being used to modify perhaps the trajectory of that disease state, then it's considered an interventional trial.
Great.
Thank you, Karen. I guess from my clinician's hat, a few things to consider are, in healthcare, how our children are typically being, tracked and managed by your providers. They're always evaluating your children. We do have our, regular interval visits where, we're trying to understand, engage the condition. We're evaluating for things that may be coming down the pipeline for our children. So we're always doing kind of new scans and different tests to try to catch new indications before they become symptomatic or become a problem, so we can intervene earlier. And then we're managing symptoms as they emerge with different treatment choices. Often in the realm of healthcare, we're only providing medications that are FDA or regulator approved. When we talk about clinical trial research, often these are non-approved medications that are still being evaluated by regulators. And what, also to your point, Morgan, you mentioned a few things that, are often embedded within interventional clinical trials as well. Often in that study, they want to really get a well-rounded view of whether that drug is working and whether it's safe. And in order to be able to manage or understand whether that drug is both safe and effective, our children will often have to have, blood tests or different scans in order to be able to truly understand the clinical and safety profile of that therapeutic.
Thank you both. That makes a lot of sense, especially the distinction between interventional and non-interventional.
So now we can go on to different types of clinical trials. So Karen, maybe you and I can kind of go back and forth on this one, but maybe you can start by talking about it from a drug developer's perspective, and then I could talk about, the clinician perspective as well.
Sure. The FDA, our regulatory body in the United States that approves drugs, expects investigational drugs or new drugs to go through specific stages of clinical trials. And the first stage is just discovery. And that's where a company would basically use a platform of some kind to discover some small molecule or some antisense oligonucleotide or develop a gene therapy for a particular disease that is of interest to them. At which point, after they've discovered this candidate drug, they will then put the drug through a number of safety tests as well as efficacy tests in animal and cell culture models, so in a petri dish. And they will then be looking for characteristics of the drug, like does it cross the blood-brain barrier in animals? Does it appear to be safe and effective in improving the state of that animal model, be it a model for example, SCN2A, or a model for some kind of seizure phenotype that they are thinking that their drug should be able to target. If those preclinical data look really good and strong, then the preclinical data is presented in a package to the FDA, and that package is then given the designation of a investigational new drug, an IND. And that IND, a designation from the FDA, allows for that company to now test that drug in human beings. The first clinical trial, or the one that involves human participants, is called a phase 1 clinical trial, in which safety is evaluated as the primary outcome usually, as well as dose responses. So in any kind of study in humans, you want to see that there is improvement that basically echoes or comes along with the increase in dose. And so that's why you would do an increase in dose in the phase 1 study. You might hear terms like SAD and MAD. That's actually not sadness and madness. It refers to single ascending dose or multiple ascending dose. And those are dose levels that you give participants to assess the safety at different doses and to assess potentially a little bit of the efficacy too at different doses. A successful phase 1 clinical trial means that that drug will now move on to a phase 2 clinical trial. That again is a small, very tailored clinical trial. It might have fairly restrictive eligibility requirements, which we'll talk about in a moment. And it's tailored to, again, look at dosing, safety, and efficacy in a small, well-defined population using a well-defined endpoint. If the results then support moving on to phase 3 now becomes what's called the pivotal trial. It is a larger study that is broadly enrolling more people, and they try to increase the number of people and the number of eligibility, the people who are eligible, so that they can have what is called a broad label, basically, so that their drug can be assigned or prescribed to a large number of people and not be, a narrow range of you must be between 6 and 8 years old in order to take this drug or something like that. So they want to enroll a large swath of people that's representative of their population, their target population, and then they'll try to show efficacy. Now, this is the efficacy endpoint for the phase 3 clinical trial. Once that result comes in, and if it's positive, then that drug can be approved. And that's what the FDA will then designate as an NDA, a new drug application, which is then submitted and based on the positive data, then gets approved. Then people can get prescribed this drug throughout the United States, and the drug can be used in clinical settings from a prescription from your doctor. Phase 4 sometimes then occurs, and that phase means that they are going to collect real-world evidence. There is no placebo control. There's no formal set of arms. There's no start date and end date. It's basically to look at how is that drug performing in those people who have now received the prescription for this drug. Is it still safe? Is it still performing with the efficacy that we would expect? And that's phase 4 post-approval. That's what those words mean.
And thank you, Karen. From the clinician standpoint, talking to a caregiver, I think the important points of consideration are, as we think about these first 2 bullets of discovery and preclinical, like those I would bucket into one group, and then phase 1, phase 2, phase 3, and phase 4 into a separate group. Phase 1, phase 2, and phase 3 are where you will actually have an opportunity to enroll in the clinical trial. And for those standpoint, as you progress from phase 1 to phase 2 to phase 3, the knowledge base that we have on the new drug and development grows. So we have a better appreciation of the safes and the potential efficacy benefit of each molecule. So when talking with your clinician on trying to gauge whether is this the right opportunity for me, Karen mentioned that phase 1 there are often kind of multiple dose changing. So at phase 1 they're really trying to zero in on what is the appropriate dose for this medication in this patient population. So enrolling in phase 1, you have to kind of go into that trial, understanding that for many molecules it may be a subtherapeutic dose, but there are often opportunities after that trial to enroll into something called an open label extension. And that open label extension often has a little bit more flexibility on being able to dose into the therapeutic range. So phase 1, they're trying to be like very, very cautious on safety. But, with that caution on safety, there is often, a lower ceiling for efficacy initially. For phase 2, often in phase 1, not so much in rare, but for many studies, phase 1 are often in healthy volunteers. And then phase 2 is often kind of the first trial within the patient population. But for rare diseases like SCN2A, often they'll kind of jump right into the patients for the phase 1, phase 2. But phase 2, they're dosing in a range that they believe is therapeutically efficacious. And the risks they're still trying to frame since it hasn't been dosed in that many patients. So there are real risks in phase 2, both from an efficacy and safety standpoint. But this is often kind of if your child's, if you desire the clinical trials are right, and it seems like a good drug that your clinician is excited about, is an opportunity to get, to grab new time. Often the time from phase 2 to an approval can be years and years. So it could be an opportunity to get your child on drug much earlier. That could be, totally transformative for the child. But understanding kind of the risk rewards of phase 2 is something that you certainly want to talk about with your clinician. That could be very drug specific. Phase 3, from clinician to caregiver's perspective, that dosing and patient population are really what the drug developer believes is appropriate for an approved drug. So at that point, they believe that regimen and how they're dosing it and how they're doing it is as close to clinically trial or close to approvable ready as possible. So we have a pretty good understanding of what the safety profile looks like for that drug. But again, phase 2 is small numbers. So phase 3, it's going to go into many more patients. So we'll get a better understanding of the profile at that point. Each of these often have an opportunity for an open label extension. So you have the opportunity to stay on drug kind of as the trial, as the drug development program advances from phase 1 to phase 2 to phase 3. If you enroll at each of these phases, you have the opportunity to stay on therapeutic until it's approved in the long run. I guess we can kind of go on to the next slide here. But talking, roughly what, we kind of mentioned here of the open label extension and why sometimes, it's appealing to enroll in an early phase trial. That's because it takes a long time to get a drug approved. These are just some kind of rough estimates here of what a drug could take to get approved. From preclinical, which programs can be in for years and years as they try to really generate proof of concept and sufficient data to support an IND submission to regulators. Once it enters phase 1, that phase 1 program is about 1.5 years on average. Phase 2 can be 2.5 years, and phase 3 can be 2.5 or more. So if you kind of add up each of these of the clinical trial program, you're at 5, 6, 6.5 years before it's approved and is commercially available. So you can see that, sometimes if the drug looks, promising, the preclinical data looks very supportive, and your clinician is excited about it, the risk reward of enrolling in a phase 1 trial and not having to wait that 6 years before it's approved, that's a lot of time that, potentially your child could be getting, very material gains in a clinical trial. And you're also supporting research that can advance this so that it can be approved, which is, really important, just for our whole ecosystem. But I think as a caregiver, knowing that you're kind of contributing to something that's often kind of bigger than yourself is often very validating and important.
I'd like to jump in and discuss why these clinical trials have to take so long. And the answer is it depends on disease progression rates, and it depends on what the endpoint that's been chosen will, how malleable it is with time. So, for example, in neurodegenerative diseases, for people, for example, who have Parkinson's disease, we know that's a very slow progressing disease. Those clinical trials, especially the phase 3 trial, tend to last 5 years. And the reason is that in order to see a remarkable difference, a change in longevity or in the motor disability that develops in Parkinson's disease over time, while it's being managed by the dopaminergic drugs that we already have on the market, it takes a long time for even an effective drug to show a statistically significant difference between the treated population and the placebo population. So that's why trials have to take a long time, because we're waiting for the drug to get into the bloodstream or up to the brain to take effect, to show an effect that's significant enough that we can statistically argue to the FDA, hey, this drug is actually working and it's improving people's lives. You need time in order for these drugs to show those kinds of effects.
Thank you, Karen.
I'd like to jump in with the caregiver hat. So understanding that, if I decide that I would want my child to participate in, let's say, a phase 2 or phase 3 trial, could you explain or elaborate? Will they for sure get the drug that's being tested, or could they fall under the placebo category? And how do I
Yeah,
be bereaved or like briefed of that?
absolutely. So if we could go to the next slide, there's a very nice illustration of
Lovely.
the typical kind of clinical trial. Now, not all clinical trials look like this, but let's, for example, look at this study design of a clinical trial, because this will answer your question, Morgan, which is an excellent one. And that is, if you decide to participate in a clinical trial, can you be certain that your child will be given active drug? The answer is no. And the reason is that for most clinical trials, in order to be valued and respected by the FDA, they need to have a placebo arm. That means you're given something that kind of either looks like, tastes like, depending on the route of administration, makes you feel like you are being administered drug, but it may not necessarily have the active component. And the reason for that is that drug companies need a untreated control in order to show that their drug is actually improving the quality of life and is efficacious in managing the disease state in the active population. Because as we know, even in SCN2A or in other types of neurodevelopmental conditions, there's a lot of variability that can occur. Some days are good days, some days are bad days. And in order to understand and tease apart, oh, overall, this child, given the active drug, is actually doing a lot better than he or she was before, you need a placebo control. And so what happens is you, Morgan, or whoever is interested in enrolling their child in a clinical trial will go through these stages. The first is to determine whether or not your child is even eligible to participate. There are eligibility requirements that essentially a clinician is going to check off, like check boxes. Are they a certain age? Are they presenting with seizures a certain way? Are they presenting with neurodevelopmental difficulties in a certain way? All of those are determined by the sponsor based on a lot of research and a lot of statistical modeling to show that if their drug is active, they have a chance at showing this over this using this kind of study design. So then you are eligible, you're screened based on those eligibility criteria. You're then randomized to one of two groups in this situation, the active drug group or the placebo group. Now, in this condition, you can see there's a 1-to-1 ratio. You can either have a 50% chance of getting onto the active or a 50% chance of getting onto placebo. Different trials can be weighted in different ways. For example, there are times when we think that it might be more advantageous to have more people on active than placebo. And we could do a 2-to-1 ratio rather than that. So you would have twice the chance of getting onto an active drug than on a placebo. And then throughout the study, you have follow-up visits to determine whether or not what the drug might be doing and whether or not it's safe and efficacious for you, and then, or your child. And then at the very end, as Sean was mentioning, you can either choose to exit the study. You can actually choose to exit the study voluntarily at any point in time. But as the trial ends, then there is a specific follow-up visit that is the end visit, or in many cases, a drug company will offer an open label extension. And that means that even if you didn't know whether or not you were on active drug or on placebo during the trial, you will now be given the chance of having active drug to try and in the open label extension. And that means, you know, everybody in there knows that they're taking active drug.
So,
Thank you,
Karen.
From the clinician's perspective, I think one really important thing to have on your checkbox when you're doing your diligence on any clinical trials is when you're going through the informed consent, One really important question is, does this trial have an open label extension? They are very, very common, but they're not guaranteed in every trial. And knowing that right off the get-go, knowing that you are guaranteed to have, drug if you want it, is an important thing to know going into that trial beforehand. And before jumping on to the next slide, I'm going to go back up to this and just add two other quick blurbs. Karen mentioned, the reasons why these are long. One is absolutely because they take a long time to mature. The other is because in rare disease, they often take a while to enroll. Rare disease drugs often require clinical trial sites that have expertise in your condition. And for rare diseases, there often aren't that many of those. So when you look at a lot of rare disease trials, there's often only a few sites that are enrolling patients for the trial. And one problem with that is, it hopefully increases the expertise around the indication, so they're better able to manage the condition in the trial. But then you limit the number of patients that can actually get to that site and enroll in the trial. And Karen mentioned all the statistical modeling in order to show statistical significance. A big part of that consideration in math is the number of patients enrolled in the trial. So, one thing that can be done to shorten these timetables is increase the speed of enrollment. And that's also a consideration when we think about us opening up multidisciplinary centers for SCN2A. As those centers then become centers of expertise that have a lot of SCN2A patients. So we're always looking for ways that we can kind of condense this developmental timeline for SCN2A. And one other way that we, as an advocacy group, push to try to make this go faster is innovative trial design. So phase 1, phase 2, and phase 3 is the classic drug development kind of program. But in rare, there are often ways to be innovative to collapse phase 1 and phase 2 into one, or to change kind of the randomization paradigms or different ways to be able to design the trial in order to speed up that path to approvability. So we're always advocating for you and trying to find ways to push the needle forward to get these drugs quicker to our community.
I'd also like to add that the drug is provided and the clinical trial care is provided free of charge to the participants. So if you do elect to go into the open label extension and know 100% that you are now receiving active drug, you should not be charged for access to that drug. That is part of what the sponsor will pay for.
Maybe one other point of note, just kind of playing the clinician role, is like the line of demarcation between things that are going on within the clinical trial and then also your clinical care that's ongoing for your child. So the clinical care, your kind of baseline will still continue to go through, us, your regular clinician. And if anything, emerges in the clinical trial, it'll still be your clinician group and your main hospital that will manage through those, anything that pops up with your child, and they'll communicate back with the sponsor in order to get any information on the drug or anything that they need in order to manage the care. But your primary care provider and your home base neurologist will still be kind of leading the charge from a care standpoint. I think that's an important thing to kind of know going into these clinical trials.
Before we move on, I have another quick question. Could you clarify from the caregiver hat, if I decide that I want my child to participate in a trial, will I know if they're receiving active drug or if they are getting placebo? Will I know that information?
You will often not know that. It's often blinded. And often in these trials, it'll be blinded on multiple levels. So not only will you not know as the clinician or as the caregiver or patient, but often the clinician that's administering the drug doesn't know who is getting treatment and getting placebo. And the reason behind that is that they're really trying to understand, often people thinking they're on drug can actually change the way that they behave. And those, subtle changes in behavior when you're trying to assess really, often minute changes in whether a drug can be beneficial or not, can be very material. So the blinding is important for us to be able to truly understand the safety and efficacy profile of the drug. There are instances where if a safety event emerges where an unblinding event can happen, but that'll often be determined by a data safety monitoring committee. That's an independent third-party committee that is in place for all clinical trials, and they're always evaluating kind of the safety profile of the drug while it's ongoing, and they make those determinations outside of the sponsor and outside of the patient or caregiver. Karen, are there any perspectives that are important from a drug developer that on that question?
I just wanted to underscore, that was a beautiful answer, Sean. I just wanted to underscore that when you see a trial being advertised as double blind, that is what it is meant. Both the participant as well as the clinician who's administering the drug do not know and whether or not it's active or placebo that the patient is receiving. And again, that's important for determining truly what the efficacy is in terms of trying to minimize a placebo effect.
Okay. So now we go on to a topic called drug modalities. And what that means is there are different types or classes of drugs that our children may or may not be eligible for. And these are kind of a few of the bigger buckets that we look at from an SCN2A standpoint. And these are small molecules, and small molecules are kind of your classic drugs that we're all used to. These come in tablet form or liquid form. These are things that you get and are regularly using on a day-by-day basis with our children. Then there are repeat dose genetic medicines. And a few of these that are out there are antisense oligos, or often referred to as ASOs, and shRNAs. These are a form of gene therapy, but what's different about them versus kind of what we typically think about with a gene therapy is these require chronic administration. So these are often kind of dosed either monthly or bi-monthly, or for shRNAs can be twice a year. And these are often kind of delivered via intrathecally or with a port that allows for kind of easier administration. And administration of these often require a specialized clinic to deliver them as well. It's often not something that you could deliver in your home or by your regular neurologist unless you're at a major academic center. Gene replacement, for a lot of our kiddos, our genes are dysfunctional. They're not working the way that they're supposed to. And for some of us, they don't even have the gene required, or at least a sufficient amount of the gene in order to be to work and function typically. So gene replacement is another form of gene therapy where they're actually replacing the gene or delivering a healthy copy, often in the form of with the delivery approach either being a virus or a lipid nanoparticle. Cell therapies, there aren't too many of these for SCN2A, but it is an emerging area of therapeutic. So we'll see what the future brings in that regard. But these are involved in delivering actually cells into your body that can have a therapeutic effect. Gene editing is another form of gene therapy. What's different about this is it actually goes into your DNA or your child's DNA and fixes kind of the root cause problem. And there are a number of different versions of gene editing that have emerged over the past decade. CRISPR is one that we hear about a lot. Talon's another one. Base and prime editing are another ones, and zinc fingers are another one. Many of these are already in preclinical development for SCN2A, which is very exciting. But these are a newer age or a newer modality of treatment that are exciting, but are still early. So the total profile of how effective these can be and how safe they are is still very much up in the air. And neuromodulation is another form. A lot of our kiddos are already receiving some form of neuromodulation. These are often implantable devices that can control how your nervous system acts and fires. And they're often able to monitor how your nervous system is functioning on a day-by-day or hour-by-hour basis and fine-tune that neuromodulation in order to prevent or decrease seizures. But important considerations with each of these are dosage, administration route, location of where you receive them, and the safety profile. Maybe Karen, from a drug developer's perspective, are there important considerations that our community should be aware of?
Simply that each of these different drug modalities will usually have slightly different trial designs based on the needs of that modality. For example, sometimes a small molecule might take a long time because you take it orally, let's say. It might take a long time to build up in significant enough levels in the bloodstream to get across the blood-brain barrier. And so that will affect the length of the trial. Repeat dose genetic medicines will, of course, require more clinical visits in which the drug is administered to your child than would, for example, the small molecule or the one-time gene replacement type of therapy. So each of these different modalities actually affect the clinical trial design. And when you're considering whether or not to participate in a clinical trial, that's one of the things to keep in mind, that there might be multiple administrations, there might be one-time administration, there might be a longer trial because of the pharmacokinetics of the small molecule involved. So there's a lot of different variables that are affected simply because of the drug modality.
Thank you, Karen. One important kind of, if you want to be fancy when you're talking with your clinician about, or even the informed consent, if you're considering clinical trials, is ask about the washout period. And the washout period it means like once you stop taking any one of these drugs, how long is the drugs in your system still effective for? And why the washout period is an important consideration for us as caregivers is if there were to be, a safety event, which is also like something that's always top of mind when you're considering these, is how long until we're kind of back to baseline. So for small molecules, the washout period is can be very short, sometimes hours or days, and these are for the drugs that a lot of our kiddos are getting, like, phenobarbital or, Keppra, these are things that wash out fairly quickly. Um, but when we start to get into different genetic medicines, the washout period can be much longer or even indefinite. And those are important considerations for, us as we're making these clinical trial decisions. For repeat dose genetic medicines such as ASOs, the washout period can be months. For shRNAs, it can be, half of a year. For gene replacement, gene therapy, and gene editing, it could be permanent. So there may not be a washout period. So it's a one-time treatment that, if successful, has, in theory, permanent therapeutic benefit, which is incredible. But in the event that something goes wrong, there are longer tailwinds of consequence. So it's important to kind of have those in mind when you're thinking through the calculus of whether this trial and this situation is right for my child at this point in time. And neuromodulation I think that's one of the benefit. While there is often a procedure required to install the neuromodulation, if there does end up being any real or perceived safety from those, they can just turn it off remotely often. So it's kind of a Bluetooth type of event where they turn off the neuromodulation, and so the washout period can be almost instantaneous.
Great.
Right. I think this is another kind of way to kind of think about our different drug modalities and what they look like. But have we touched on most of the elements from Morgan? Are there any caregiver perspectives that you think are important for us to touch on?
Yeah, I guess just when you brought up the washout period, let's say I enrolled my child in a gene editing trial and they were given a one-time dose of a CRISPR modality. Given further scientific development and technology, would that mean that they could be ineligible from ever participating in an additional trial down the line if they did receive a CRISPR injection? Or
Yeah, a really great question. And I'll speak about it from the clinician standpoint, but then Karen, maybe you can talk about the technical reasons of why they may become excluded for future clinical trials. So yes, there are certainly certainly times where, you know, being enrolled in one type of clinical trial will prevent you from being enrolled in another type of clinical trial. Particularly at the exact same time. That's often almost a guaranteed no-no of being in two clinical trials at the simultaneous time. But there are instances where, being treated with one gene therapy can prevent you from getting a future gene therapy. And one of those reasons why is, often gene therapies are delivered with a virus. And there are a number of different viruses, but the most common are called AAV. And these are a very well-known safety profile, and they're often non-integrating, so their safety profile is much safer. But upon dosing with these viruses, in order to deliver, the payload, which is, either a gene replacement or some form of gene editing, you get all this virus at once. They go into the cells they need and make the correction that they need, but the body generates a very strong immune response to that virus. So it kicks out all the virus, and then your body's kind of primed and ready to go. So if it ever sees a virus again, it's gonna, fight that off right away. And why that's important for future is if a future gene therapy comes out and it's using that same AAV, if they were to try to deliver that, your body's ready for it. It's already seen that virus, so it's gonna kick out that virus even before it has a chance to be therapeutically beneficial. There are a lot of work being done to think through ways to be able to allow for repeat dose gene therapies, but they're still pretty early days. So from a safety and a safety standpoint, that's an important consideration. But Karen, there are important considerations from a drug developer standpoint and the risks of trial design and trying to maximize their probability of success that would maybe change how their eligibility criteria are.
Yes, that's absolutely right. Just to reiterate, actually, what you said, Sean, there are things that are called drug-drug interactions, and there's also this issue, especially raised in, other clinical trials of gene editing, where we see a pretty significant neuroinflammatory response. And so, and that's the immune response that Sean was referring to. When you have a dose of virus entering the body, the immune system learns, oh, this is foreign, and we're going to fight it. And then the next time you're exposed to that, the immune response is even more active and violent. And that can actually lead to some safety concerns. And so, of course, what drug developers are most concerned about is safety, safety, safety. And if we think that there might be the chance that a certain gene editor that or viral capsid that you've been exposed to once before might give rise to this inflammatory response again, that is a big safety issue. And sponsors are going to put down in their eligibility requirements that you must not have been exposed to this in the past for your own safety.
Thank you, Karen. I think one other point just to mention too is, Karen mentioned early on about understanding disease progression. And it's super important for, designing an appropriate clinical trial is, you have an understanding of what disease progression typically looks like without any kind of novel interventions involved. And sometimes when you're enrolled in a clinical trial that has, potential of altering disease progression in a way that we don't know about. So if all of a sudden you're no longer going to be a representative example of what a typical disease progression looks like in this indication, that's also a consideration of why you could potentially be excluded from a future clinical trial as well. Good question, Morgan. All right. I think we did touch on a few of these, but I think this is a really nice figure of kind of really driving home the differences between vector-based gene therapies, ASOs, and small molecules. And then I can't see it because my share screen is a little bit goofy. Let me pull it up on the screen here. Ease of delivery, and then how long kind of that washout period is. So for kind of starting at the bottom, small molecules are very easy to deliver. Small molecules are able to kind of get to all areas of your body, often easily crossing the blood-brain barrier to be able to get within the deep regions of your brain. But then they also have very quick washout periods or half-lives. ASOs are harder to deliver, often requiring intrathecal injection with a longer washout period, weeks to months. And then vector-based gene therapies or gene editing with each of the modalities that we talked about, gene replacement, CRISPR, and then tRNA suppression. The ease of delivery is harder, delivering, a big payload of virus either either intrathecally or IV, depending on the virus, or sometimes they even have to deliver directly to the brain. So a much harder means of delivery, but these remain in your body lifelong because the changes are permanent. Any other things that are important to reference on this slide from the team? All right.
Thank you.
So clinical trial design, eligibility, and enrollment. We've talked about this kind of throughout our entire talk here, but we'll kind of get into the weeds on inclusion and exclusion criteria, informed consent, study endpoints and metrics, and trial locations. And I think that Karen is going to tackle some of these just from the drug developer standpoint.
Sure. So the drug developer is very concerned about designing as successful a clinical trial as they possibly can. And by successful, I mean both safe for the participants as well as efficacious, a demonstration of efficaciousness of their drug. So a lot of times it might feel really frustrating to look at the inclusion and exclusion criteria of a clinical trial and say, oh, why are they being so particular? Why does my child have to have at least 4 seizures per month in order to be enrolled? The reason is that the clinical trial has been, as I said before, statistically modeled based on prior data, based on natural history data, for their statistics to understand how many people they need and what type of presentation those people have to have in order for the drug to demonstrate what it can do. For example, if I designed a clinical trial for a seizure, for an anti-seizure medication, a new anti-seizure medication that should lower the number of seizures dramatically, I want to start with a population that I know is going to have seizures within a certain period of time. Because if my population that I enroll don't have seizures or suddenly have seizures that are under control that are not due to the drug, now I've diluted the possible effect that I could see the drug having. Because if a drug just gets rid of 1 seizure because your child just had 1 seizure in the 6 months, that's very different than the drug actually being able to demonstrate it took a child that was having 600 seizures a day down to 2. And statistically, I have to be able to understand what I need in terms of inclusion and exclusion criteria in order to meet that high standard of showing efficacy for the drug. So that's why inclusion and exclusion criteria exist. They also exist, of course, because of safety. There are certain types of drugs that might create an immune response in certain types of people who have been exposed, let's say, to another type of CRISPR or to a type of anti-epileptic drug. And so we'll say, please, if you've taken this drug before, you're not going to be included in this trial because we know it's a safety issue. We know there's a drug-drug interaction. And so that's why there's an exclusion criteria on that. So actually, exclusion criteria are very carefully chosen and carefully designed. A sponsor wants to include as many possible people in their trial as they can, because as Sean mentioned, enrollment can be quite a challenge, especially in a rare disease. So there's the tug and there's a push and pull of wanting to include as many people as possible, but at the same time, observing specific safety standards as well as the statistical standards that we have to meet in order to design a potentially successful trial. What's also very important, of course, is informed consent. We want, as a sponsor, people who voluntarily sign up to participate in a clinical trial to understand what the risks and benefits are of participating in the trial. There are certain commitments that the sponsor will make to the participant, such as we are definitely interested in your safety throughout this trial, but there's also things that every individual participant has to make for their own comfort and their own risk level, and that is a very personal decision. That personal decision can be helped and aided by what's called the informed consent form, where the sponsor will actually lay out what we think are the benefits and risks that we have seen from studying this drug. The study endpoints, as I mentioned before, are extremely important for the sponsor to choose the correct endpoint. Choosing an endpoint that the FDA recognizes is an established, validated endpoint is the way that the trial is going to be deemed a success, not by the sponsor, but by the FDA. And that is really the metric that sponsors have to obey. So if we went out there and chose a very, very different endpoint that the FDA does not know or recognize as a validated endpoint, the trial will automatically not be regarded by the FDA as something that we could submit for eventual approval. And that is a great danger. So not only do we take into account what the patient voice says is important, like we want, we want a trial or we want a drug that actually helps us control seizures or improves quality of life by making the child more mobile or more communicative. We definitely take that patient voice into account. But if there's no really good validated measure for measuring, for example, a specific type of cognition that matters to the patients, we're going to have to either develop that endpoint or we're going to have to use a different kind of endpoint that is recognized by the FDA that could be a surrogate measure for that measure of cognition, for example. And then, of course, we choose trial locations very carefully. We choose them on where we think we can enroll, and we also choose them based on the expertise of the clinical team that are there, because as Sean mentioned, ASOs, CRISPR, they require specific types of experience and clinical trial readiness and expertise on the part of the clinical team that are actually dosing and delivering the drug to participants. And so we need experts that understand drug modality, how to deliver, how to track the progression of the disease, familiarity with the presentation of the disease, things like that. So that if a safety event were to occur, that clinical team would actually recognize it as something that needs to be followed up and something that needs potential immediate unblinding.
From a clinician standpoint, I think one important recommendation I would make, to the caregiver or patient is, on the trial location is ask, like, what is the degree of expertise you have in this modality? Have you run other clinical trials successfully with this class of medication? Have you dosed patients like this? If so, how many? If the answer is this is our first time, it probably is worth kind of doing another layer of scrutiny and talking about it a little bit deeper with your clinician of, I know this is a little bit closer, but is this the best facility for, a potentially high risk intervention? Is there one maybe an hour or two further that has a lot more experience? Those should be kind of included in kind of your risk calculus of thinking through not only is this drug and trial appropriate for me, but is this location the best one for me too? On to participation and data collection. So just a few elements here, and then we're going to jump to a slide that will kind of help us think through what participation looks like in a clinical trial. But conceptually, what a clinical trial participation looks like, there are logistics, which are really big. When we have a rare disease kiddo that's maybe in a wheelchair, or if you've never traveled with them before, thinking through the logistics of how long you're going to be there, the things you're going to need. Do you have oxygen? Do you have your drugs ready? Your baseline medications at a pharmacy close. All the blocking and tackling that we do to manage our kids on a day-by-day basis. Is that all? Have you considered each of those in kind of what's required for the clinical trial? Travel, whether the sponsor will help support financially and actually the logistics of traveling for you. They should to some degree, but not cover everything, because then it can be a level of coercion. Um, the reimbursement and accommodation that they provide. Doctor visits. So doctor visits, I guess, at a high level, when you enroll in a clinical trial, you'll be getting a medication, but then, they have to be evaluating your trial to see if it's both exams, there'll be often blood tests. So for our kiddos, which are hard sticks, another important question is like, who's going to be doing my blood test? Or do I want the best there? I want the A team to do our blood draws. I don't want my kid to be a pincushion. Vital signs and imaging. Ask about if all these are going to be done in one building, or are we going to be traveling all over a hospital to get all these done? Make sure you're comfortable with what that process looks like, because it's a lot. But there are ways that a sponsor can design the clinical trial experience to be better for you. And if your experience is poor, that's why we're here too as a patient advocacy group. Communicate that back to us. We had a bad or good experience, and we can relay that back to different sponsors and saying like, this was really good. Really appreciate you doing this, or maybe there's opportunity to tweak the protocol a little bit to make it easier on our kiddos. But rating scales. So Karen mentioned the different ways that the FDA looks at whether a drug can be approved or not. These are just a few scales. I know all of us have done a million Vinelands over our time, so we're bleeding Vinelands, but there are other scales too that they ask questions and we give them feedback on how our kiddos and how we are doing as caregivers as well. Data collection and compilation. So different ways that they capture data. So seizure diaries are very common in our types of clinical trials. And it's important to be consistent in those diaries, because those are going to be kind of the meat and potatoes of make the determination of whether a trial is successful or not. Data blinding. Karen, talked about that at length so far, but it's an important kind of consideration for the caregiver and the clinical trial experience. And confidentiality during a trial participation is super, super, super important. So even though you're in a clinical trial, it's not appropriate to be talking about your experience in the clinical trial and whether you're having a benefit or not publicly, because that can change the perception on whether a drug is good or bad. And then that can change the placebo effect sizes in the trial. And, there have been plenty of instances in other clinical trials where, a placebo effect size. So these are patients that haven't received any drug at all, but the effect size in that arm of the trial was abnormally large. And then it made it seem like, a drug that could have been beneficial wasn't statistically significantly beneficial because the placebo effect size was so large. So maintaining that level of confidentiality, and whether the drug is working or not, and is it safe or not, it's important to kind of keep that between you and your clinician, and your neurologist, your home base, and not be talking about it very publicly with the community. But the next slides will kind of get into the blocking and tackling, what maybe a true experience would look like. And I'll leave it to Morgan to kind of talk through as maybe a pseudo patient or caregiver that went through a clinical trial, what your experiments could look like.
Yeah, for sure. Thanks, Sean. Yeah, so hypothetically, I will be playing the sibling managing the care of my brother who has a SCN2A gain of function mutation. And really, to kind of hone in all the points that we have talked about thus far, to even start this conversation on their participation in a clinical trial, it came down to me talking with his primary care provider. And as Sean and Karen mentioned, it's a really personable conversation where you have to manage the risks, benefits, really look at what my day-to-day care with him has been like. And so you kind of see here, starting at the upper top left corner, that's where that all goes down, essentially. We had decided that seizure control was one of the main things we really wanted to try and help for my sibling. And we had decided that let's see if there's any eligible novel anti-seizure medicine trials out there that he could potentially benefit from after being already on 5, 6 anti-seizure medicines. Is there a phase 3 or a phase 2 trial that could potentially invoke some more benefit than what we're already seeing. And talking with his healthcare provider, we really decided like seizure control was what we wanted to try and focus on. So I worked with his HCP. It was a couple weeks long when we found a trial that his provider potentially thought he could be eligible for. So that's when we kind of got into the screening and the eligibility part of it, where Sean and Karen mentioned. You go through all of the inclusion and exclusion criteria. My healthcare provider helped us do that. I got the contact for the sponsor. It was their clinical research coordinator. It was about 3 to 5 weeks of just screening to really see if my brother could be actually a viable participant in this trial. The trial was a small molecule, so we were briefed on the double-blind, placebo-controlled, all of those things. And once he was deemed eligible, we signed the informed consent. And so that's when we got to have those really personable conversations as well on things that were important to me as a caregiver and for him. Where's the trial site going to be located? How much travel am I going to have to do? Has this drug been tested in a lot of patients like my brother before? We had a lot of those conversations. And fortunately, my primary neurologist did not host the clinical trial at their site. So we had to travel 3 hours north to a major metropolitan city where the trial took place. And the sponsor did provide accommodations and reimbursement for those types of things. So once informed consent was signed, I was briefed on where I was going to have to go and what I needed to do. The study treatment started. So you can kind of see here, the inpatient visits are marked with the little black diamonds, and then like the up titration, that's kind of when you start the dosing of the medication, is in the green, and then the down titration is in the red. I was told that this would be a 15-week small molecule trial, so I'd have to go in every other week and bring my brother to the primary site, which again was a drive 2 hours north of where I currently live. And we went through and did this trial. So the first visit was more where you get the baseline of the physical exam and the blood tests, seizure counts. 4 weeks prior to this first patient visit was really important. I had brought my brother in, and that's where he got his blood drawn, all of the other things that Sean and Karen had mentioned as well. And then I was sent home. So this particular drug was dosed twice a day, and I was trained or briefed on how to actually administer it, because I wouldn't be going into the doctor's office every time I had to give the drug. And so you can kind of see it follows this 15-week cycle where every other week I'll be bringing my brother up to the clinical trial site to do the full physical exam, the full blood draw. But then every single day when I'm at home, kind of going down to this third bar, is where I'll be tracking his daily seizure count. As I mentioned, what we really wanted to focus on was seizure control for him. So we had to count the seizures every single day using a thing called the seizure diary, which Sean had talked about. Most commonly now it's an e-diary. So I was briefed on how to do this, and I had to count and control all the seizures that my brother was having every single day. And I would do that coinciding with the appointments that I would be going to at the actual primary office, which was the 2-hour travel. And so I would say the experience was great, because I knew that there could be a potential benefit for my brother, and I was there participating with him in real time. But it was a lot. I would say it was a lot to travel, a lot to count and kind of be on guard for. But the whole overall experience was great, and I always had support from the sponsor's contact and my primary care provider. So if there was an emergency, I made sure to contact my HCP or his HCP directly, and we would handle that. But that's exactly what it was like to participate. And then at the end of the 15 weeks, we were given the option if we'd like to continue into the open label extension, bringing in what Sean and Karen mentioned before. I didn't know during this whole time if my brother was receiving active drug or placebo, so we found it at the end of 15 weeks. Now I can enroll him in the open label extension, and I will now know that he's actually getting active drug. And then from there, that can be a couple months to a year, depending on how it goes. And I want to also reiterate that I could not express like any of my feelings or things I had noticed while participating in this trial. Like, let's just say I were to actually notice that my brother did have a reduction in seizures each day, I could not post about it on Facebook and let everybody else know. I had to really maintain that confidentiality that Sean had reiterated before. But as you can see, the whole process took a couple months. 15 weeks is a good 3 to 4 months. But I had support with the sponsor accommodations, and again, just really maintained communication with his primary care doctor. And that is kind of how it will look on a broader scale. Also honing in on what Karen said, that the type of drug modality really can influence trial design. Oh, my dog's barking, sorry. Yeah.
Thanks, Morgan.
Yeah.
Well, one thing I want to highlight too is that the daily seizure diary. Morgan's a situation where, she's taking her brother. For many of us, like, we're not the only people that are helping, or, parents or in-laws. Keeping everybody abreast on the daily seizure diary protocols is important. So you have consistency throughout the day, that you're tracking and logging that information on a consistent basis for your patients. So it's often not just kind of you and your kiddo, but it's often kind of your whole team has to kind of commit to these experiences as well. But thank you for going through that, Morgan. I think that's very helpful. So yeah, I think that we're almost at the point of kind of concluding, but we just wanted to highlight that we heard the survey from Leah indicating that this is a priority for our community. We're going to have a whole slew of town halls coming out about active clinical trials that are ongoing, opportunities, and different resources that we can provide to you to help you think through your goals of care, think through which clinical trials are available, which ones that are appropriate for your children. So we're excited to be able to be giving each of these to you in the very near term. And yeah, feel free to read. That's what we're here for. Please reach out to any of us for any questions, and keep your healthcare provider in the loop on any decisions or considerations you're making. They're critical for kind of framing your goals of care for your kiddo. And then we'll open up the door for Q&A.